what can you look for? Yeah.
Serine, triosine,
Right.
tyrosine, cysteine, and histidine.
I think cysteine is not extremely polar.
Okay.
So sulfur, but it's not apolar. That's not what I wanted to say. But it's not one of the really prominent ones. So sulfur is below here, so it's not that. Yeah, but I'm missing two. Yeah.
Asparagine and glutamate.
Right. Right. Okay, and again, it's worthwhile to remember the names in one letter code. Okay, so yeah, otherwise sometimes we group by other things, like for example, there are bulky ones and less bulky ones, things like that. So we don't expect you to be able to draw an amino acid, but if you hear a name, it's worthwhile to know, okay, that's a bulky one, that's a polar one, that's a whatever. Okay, you've heard probably lots about how they react and how they form peptides. Again, the important thing to remember for 3D is that this is a plane. So it's only relevant to turn around this, and here nothing can happen. Yeah, that was for the computer scientists, how we write down proteins. You've heard about the different structure levels. Yeah, I'm sure you've heard about alpha helices lots of times. Would you able to be able to build one? I found it's the difference between knowing what it's like and building one is, even for a chemist, there is a difference. But what I also found interesting is while I built that, I first built the chain and then I made the H bonds. And that was really interesting to see the difference in stability. So as I'm going to give this round, feel free to break a bond, but you do me a favor if you make it afterwards. And what you can also nicely see when you pass this around is also that really the amino acids stick out. So I think hands-on is a lot better. Unfortunately, it's extremely expensive to get kits like that. But if you have any friend who's a chemist or anything who's got a kit, I can only recommend to really play around with this, because it's the difference between knowing this and just doing it. Yeah. I also tried to build one side which is more hydrophobic and one side which is more hydrophilic. Yeah, because that's also something you often observe in structures, and it's something that helps you try to work out what the structure is like. Because if you can spot patterns like you've got a hydrophilic amino acid every fourth position or something like that, then you can say, oh, this is probably a helix. So this helps, for example, in secondary structure prediction. Okay, the other thing is, of course, you have beta sheets, and there you find other patterns. So what kind of pattern would you find in, so I said in helix, you find something like every third, every fourth amino acid has similar properties. What would you find in a helix, in a beta sheet? Where do the residues point in beta sheets? Which residues might have similar properties and dissimilar? So this is a very, very simple sheet because you've only got glycine and alanine. But you see, for example, that here the allos always point in the same direction. So every second residue, so for example, if you had a sheet at the outside of a protein, then every second residue would be hydrophilic, and every second residue would be hydrophobic and look inside. And again, this is a pattern that helps you predict secondary structure. So it's nothing that you kind of put in as a rule, but it would be something that your machine learning algorithm might learn. Okay, so if we look at structures, you of course know that we rarely look at like all atoms, because even if you're used to this kind of thing, this is the mass. We usually look at the C-alpha trace and then we try to spot the secondary structure elements. Here they're highlighted. And yeah, this is kind of focusing down on them. Again, you see the structures, the H bonds, etc. You see the H bonds here. You've of course heard about Ramachandran and the angles that you can have. Yeah, so another thing to keep in mind when we think about proteins and protein structure is that in order to stabilize things like tertiary structure and quaternary structure in some proteins, but not in all, we can have cysteine bonds and disulfide bridges. Why did I say in some and not in every? Why can some kinds of proteins not have disulfide bridges? Yeah.
Because they are not in the AR membrane, and the sulfide bridges are made in the AR membrane.
It's not just a question of where they're made. Yeah.
Maybe they are in an environment where this oxidation cannot...
Right. Some are in reducing environments, and there you can't have a disulfide bridge because it's just going to be reduced again. So having disulfide bridges can, on the other hand, tell you something about localization. Okay, Marco made a nice set of slides. Marco Punta was in our group for a while, and he's now at PFAM. He made a nice set of slides about models and pictures, etc. So just to remind you that when I'm showing you this, when I'm showing you pictures of the proteins, the question, what kind of information do those pictures have? And for example, what are you seeing here? So what is that?
A tree?
Yeah, it looks a bit like a tree.
Like one of
The name this was published under kind of is houses. So there's a way of seeing houses like this. The other thing is, this is again a picture by Goodsell. We can think of where is this, and also what is this? What are you seeing here? Well, I don't expect you really to be able to work it out.
The yellow lines are the cytoskeleton.
Yeah.
And the green thing is the membrane.
Yeah.
The things that go out are those. Is this a bacterial cell?
Right, it's a bacterium. Actually, a very small one. Where it's
There are ribosomes.
So it's Mycoplasma genitalium. So it's as I found out from Wikipedia, it's not the smallest genome anymore. They thought for a while it was, but it's the smallest that can constitute a cell, so that can kind of do it all by itself. And yeah, you in the slides online, you also have kind of the solution to what is what. But one interesting thing about this is, usually when a cell is depicted in a textbook, they try to explain what the different kinds of things are, and cells usually look extremely empty. Whereas the kind of pictures that Goodsell draws, this is also one, they try to make you aware that a cell is in reality filled with stuff. So don't let yourself be misled by pictures. When you think of a cell, it's not empty. There's lots and lots in there. Okay. Yeah, the other thing is you can draw it like this or draw it like that. Of course, you don't just have tubes here, but it helps you. This picture helps you work out what is the general arrangement of things, and the other picture helps you to work out chemical detail of how do things interact. This is another way of different ways of showing a molecule, and especially this one shows that actually this acts as a homodimer. This is, by the way, something you have to be careful with when you look at structures from PDB. Very often you only see the unit structure, which helps you make up the crystal. So it's the one thing that is repetitive in the crystal. But if something is a dimer in reality, in biological life, it could be that by some geometric operation, the crystal is just composed of the two things, and when you work out the structure, when you determine the structure experimentally, you just kind of see one part of this and all the rest is hidden by geometric operations. Then the crystallographer has often deposited the structure in the monomeric form. And in order to really work out how this thing works, it's good to look at the biological unit. I think nowadays PDB is moving more towards really pushing the knowledge about the biological unit. But in the past, you would often only find the unit cell in the PDB file. Okay. And yeah, which is this. Yeah, I think you can't guess. No, not at all. It's called the dynamics of a soccer player. And the point where we have it here is because, I mean, this is a snapshot of soccer players, but a different aspect of what soccer players are is contained in here, because they're not just here, they're kind of all over the place. And just in the same way, a protein is often not just here, but it's got parts that are moving. And for example, this loop is kind of all over the place. So this is another aspect about when you see a picture of something, think about what it shows you. And when you show you a picture of a protein, we very often only show you a snapshot. And in the alpha helix, you could also see what parts you can move, etc. So the thing in the middle is probably pretty rigid, and it's probably in the point where we show it to you, but the stuff outside can be very flexible. And also, when I talk about protein structure prediction, I always wonder why people are so specific about modeling the loops in the right way. Of course, there's some probability of having it here or there, and it might be more probable to have it in this form than in that form. But I wouldn't worry so much about the things that are flexible, but I think it makes much more sense to show the diversity of what is possible than getting exactly the snapshot you get in a crystal, right? Okay, so maybe we make a short break then after 5 minutes I'll tell you the rest, which is not going to be extremely long. Or do you want to just finish early? Just finish early? Okay. Okay, so another thing to keep in mind is that proteins can be seen as made up of domains, and now there are different definitions of domains. One definition comes from the structure, where you just say, okay, a domain is something that folds by itself. Another definition of domain is something that has a function together, or is inherited together. And an interesting question was, can I see a domain as always being an intron? And basically, to cut it short, the question is more or less no. So what you can see is that sometimes you can merge domains into one. And sometimes a domain can reoccur somewhere and sometimes you can have insertions into a domain. And yeah, if you have multiple alignments, you can often find back some parts. And for example, this part here is repeated a number of times in there. And Burkhard also tried to have a project about guessing domains just from the sequence. So he would say, okay, you've got this piece of sequence which is repeated in these proteins. You've got this piece of sequence which is repeated in these proteins. So because this is a chunk that is repeated and this is a chunk that is repeated, we think that these are domains. And the dream was that it would always be like this. So you would always be able to separate. But unfortunately, this is more the reality. You have smaller parts that can kind of be combined together somehow, and you can not always split that tidily. Okay, and this is just an interesting thing to look at. Different ways of defining domains come also to different views on how big the domains are, etc. So Prodome, Domus, Mars, COP, etc. are all databases that collect domains, and they apparently all have their own specific view on that. So when you talk about domains, you should always think about, okay, how do they define it? What is the question you ask? And what kind of definition of domain is going to help you most answer your question? Okay, some trivia about proteins. How many do we have in human? 20 to 25,000. How long are they? Yeah.
I think the mean is about 300.
Yeah, the median is around 400.
Okay.
But maybe, mean and median can be different. So do they consist of units? Of course, most of them have more than 2 domains. So if you do some statistics, you find that usually they have more than 2. Are longer proteins rarer than short ones? Yes, they are. So Booker did some statistics there. Are prokaryotic proteins shorter than eukaryotic? And he also did some statistics and found that. So there was a distribution of what is the number of residues in proteins, and the different things for archaea, prokaryote, and eukaryotes. And especially in the part where you have longer sequences, you see that archaea and prokaryotes reach, so you've covered more or less all proteins by the time you reach 1,000, whereas in eukaryotes you get longer proteins. Okay, so this is what I talked about today. So we had organism genes, amino acids, protein structures, bonds and energies, and I think I'll leave it at that. If you have questions.